some major classifications of genetics. As we know, the genetics is now been classified into 4 major categories, which are transmission genetics, where the mode of gene transmission from generation to generation is in fact is studied and also being examined. And the second area, major area of genetics, is the molecular genetics, where one studies the gene structure and the function of a given gene that may be predisposing an individual either to a specific phenotype or to the genetic diseases. The third area of genetics which is being studied is population genetics, where one looks at the gene behavior in a specific given population. So, we can go through examples under each category to tell that how the mode of gene transmission from generation to generation in fact takes place. As we know of now, the number of genetic diseases are in fact transmitted from one generation to another, that is from father to children to grandchildren and so on and so forth. And some of these genetic disorders which have stably inherited in a given family is how the study of genetics is performed in the area of transmission genetics. And there are some classical examples. If you take for example the cancer, in the field of cancer, approximately 10 to 12% of the cancer is in fact is transmitted through generation to generation. Or even if you take for example the complex diseases such as diabetes or even cardiovascular diseases are some of those diseases which are in fact transmitted from generation to generation. The study of how it is being in fact is transmitted from one generation to another, which need not necessarily limited to diseases, but also may be related to the traits or the phenotypes which we call them, is in fact is a transmission genetics. Equally important is the molecular genetics. The molecular genetics looks at various changes that in fact have occurred in a given gene, and that is in fact is responsible for a given disease, given condition and or for a given trait, and that is exactly how the molecular genetics looks at and studies at it, because any defect in a given gene in fact has a distinct effect on its function, and this altered function may lead to predisposition of an individual or even cause an individual a specific genetic disease. Population genetics on the other hand primarily looks at the given the gene behavior within a population, wherein one looks at the distribution and the susceptibility of a specific diseases within a population. In other words, there are number of diseases which in fact are other than the genes which may be involved, in fact responsible for these diseases, and which are environment related. And these environment related disorders are in fact studied within a population, wherein the gene acts as kind of a responsible factor for being able to interact with the environment and thereby manifest a given condition on the diseases. There are other aspects of genetic diseases which we call it as oncogenetics. By the very name indicates, oncogenetics means that those genetic principles which are applied to oncology or in the field of cancer. More recently, the pharmacogenetics or the pharmacogenomics has been an intense subject of discussion, research as well as translation into the clinics, wherein one looks at primarily how the drugs are in fact are affecting the genetic makeup of an individual and thereby bring about the personalized treatment or personalized medicine in a given individual. Precisely how an individuals in fact respond to a given drug or individuals who respond to specific given drug is determined by the genetic composition or the genomic composition of an individual, and thereby one will be able to treat the individual on a personalized basis. This is exactly what happens if you go to a tailor who can stitch your shirt based on your size or your height or your breadth, and similarly the drugs can be custom made or tailor made depending on the genetic composition or the genomic composition of an individual. One such example which is shown here, where you have a large population of individuals as has been shown on the top of the figure, which comprises of large population of individuals with multiple type of populations and multiple type of individuals. Majority of them would respond to a given drug, and the small proportion of it who do not respond to the given drug are in fact taken off, because they will be responders or toxicity of the drug will be detrimental to the health, and that is why the large population, smaller groups of people will be taken away for a different kind of treatment. Therefore, the population can be stratified into two groups, a major group of which can be treated with a given drug. However, individuals with a specific makeup of their genome or the genetic composition can be changed in such a way that they can be treated differently as opposed to the common population. That is why the pharmacogenomics, which uses the sequence information to measure, to predict the reaction of an individual to a given drug, in fact which has led to what we now define as personalized drug or faster clinical trials or even less side effects. As you know that the side effects are the major cause of in fact toxicity which happens in an individual, and even the clinical trials which take approximately about 7 to 8 years to complete cost more than $700 to $800 million to bring a new drug into the clinic. Pharmacogenomics is hoped that it will take care of some of these issues, so that the drugs can enter into the clinical practice sooner and even at a lesser cost than what is being happening right now. Genetics in general has its repercussions in almost every single field which we know of. It is in the field of medicine, in the field of agriculture, in the field of ecology, and in the field of law, sociology and philosophy has all been influenced by the principles and practices of genetics. Today, if we are seeing advances in the field of medicine, it is because of the understanding which has happened over last about 20 to 25 years in the field of genetics, that has been continuously translated into the clinics, so that the patients and the individuals are benefited from the advances that has been happening in the field of genetics. For example, the gene therapy principles which are now being practiced has borne out of the fundamentals of genetics. Secondly, the stem cell based therapeutics that are in fact are happening are coming out of the advances that have happened in the field of genetics, has shown a lot of promise to be able to cure a number of these genetic disorders, and in some cases indeed it has become quite successful in being able to implement and bring about appropriate cure to the patients or to the individuals. We will not go deep into the influence of genetics either in the field of agriculture or ecology or in the field of law or sociology or philosophy. However, it is very clear that the classical genetics has immensely helped in alleviating what we call as a shortage of food in India, especially after Green Revolution, wherein the classical genetic breeding of the plant has made India self-sufficient to be able to take care of their issues that related to the food security which comes from the advances in agriculture. Today, if you have crops which are pest resistant, which are drought resistant or higher yielding varieties, are in fact coming from the advances that has come in the field of genetics. With respect to law, the genetics has immensely helped in several areas, and these include in the area of forensic science, wherein one would be able to identify the criminals who will undertake the crimes, and by using the genetic principles one will be able to trace the DNA which is left out at the scene of the crime, and then being able to identify the crime and the individuals who have undertaken these crimes. Similarly, there are several philosophical and sociological issues which are associated with the practicing of genetics in real life or in the clinics, and these remains to be answered, including how the genetic information is being undertaken ethically or being practiced safely in a way that is not going to alter the major human ethics and values that is being practiced in the society and community. So, appropriate ethical rules and regulations and the control by the government rules and regulations are in place to protect the individuals, so that these genetic advanced knowledge can be practiced in an ethical fashion without any physiological or sociological barriers. Here is an example of what we have understood from some of the recent advances that have happened in the field of genetics. For example, an enzyme which is peptide deformylase, which has been identified and characterized, has so many number of properties, which is in fact is making it very useful to see that every single molecule within them in fact harbors the wonders of so many other units of life which harbors within a given life. This particular protein has more than 12 different variants, and each of these variants have in fact have a specific and function to be able to take care in a given condition. Here is yet another example of how the genetics has been identified using the forensic principles that has been demonstrated in the stem cell transplantation and making of dolly. As you know that the dolly has already been made through the stem cell transfer, and in order to obtain the concrete evidence to show that a dolly has in fact indeed made from the stem cell transfers, it needs to be proved based on the certain principles, especially the distribution of genetic material, DNA material, as has been shown here. What is shown in the figures is lane 1, 2, 3, 4, 5, 6, 7 and all of those are the DNA samples which are derived from number of other lambs which are in the farm. However, U stands for the udder, C cell stands for the cells which are used for the transplant, and D stands for the dolly, where the DNA has been extracted and fingerprinting has been done for all of these lambs that has been shown here. The dark band which you see is nothing but the specific set of DNA bands which you see, which are manifested, and as you can see, U, C and D lanes are looking almost identical, suggesting indeed that a dolly has come from the stem cells transplant and coming from the cells and the udders which have been used for making of the dolly. However, it does not relate to the other lambs as has been shown in the numbers 1, 2, 3, 4, 5, 6 and so on and so forth, suggesting that the genetic principles as we understand from the forensic genetics can be successfully used to be able to identify the lineage of a given experiment, or in this case as has been explained in the case of dolly. Here is yet another example of how one can trace the origin of a specific background of a given crop. In this case, it is the basmati rice, as has been described here in this slide, which has been demonstrated by CDFD in Hyderabad, Center for DNA Fingerprinting in Hyderabad. Yet another example of how fingerprinting can be used to be able to identify, understand the beneficial effects of those plants which are of economic value. In this case, number of basmati rice plants are in fact DNA is extracted, and to be able to identify the ancestor of this plant is in fact is run on a specific experiments, and the series of bands which you see on this particular picture are representative of DNA sequences, which are in fact quite helpful in being able to see the evolution, origin, and how long it has been evolved in this earth and is being used by the mankind for the various nutrition purposes. So, in general, genetic constitution in fact is responsible for every single activity that happens in our day to day life, which includes various activities which we see, such as during the pregnancy, lactation or exercise or sunlight or pressures or various diseases or infections, or even the circadian rhythm and even the liver functions, renal functions are all controlled by the genetic constitution of an individual, and there is an intimate relationship between the genetic constitution and the environmental factors which in fact play a major role in the disposition of an individual to specific phenotypes or the traits. You all have already know the anatomy of a given cell, and that has been shown here in the form of peak inside an eukaryotic cell, where you see the major organelles that are in fact are present in a given cell, which include nucleus in the middle, nucleolus, as well as endoplasmic reticulum and Golgi bodies and the mitochondria, which you can see in this particular slide, in this particular picture, where they are compartmentalized in such a way that they are involved in the energy metabolism, transport, storage of information and transmission of information, maintenance of architecture of the cells are all governed by various organelles and the proteins that are present in a given cell. The central dogma in fact states that the RNA comes from DNA, and RNA in fact makes the protein, and which in turn is distributed in nucleus as well as in the cytoplasm of a given cell. The DNA consists of genes, and the genes are within the chromosomes, and all of them are in fact are sitting in the nucleus, and the mitochondria, which is in fact also the way the DNA is present, is in the cytoplasm, and thereby providing a diverse pool of DNA or the nucleotide sequences which are in fact is present in a given cell. The specific processes such as reverse transcription, in fact which happens in some of the viruses, such as those viruses as HIV, which is in fact an infectious organism to the humans, and the specific reactions wherein one can convert RNA into the DNA also happens at a very low intensity in the human cell, and thereby providing a wide array of mechanisms that may be responsible for maintaining the basic units such as DNA, RNA and protein within a given cell. DNA is in fact consists of number of regions such as exons and introns, and then which come together in the form of after the splicing reaction as RNAs. Exons are the coding sequences, introns are non-coding sequences which after splicing in fact come together to make an mRNA, and these mRNA are subsequently translated into different kinds of proteins and even have several post-translational modifications in such a way that they may have even different functions. The human genome, which has opened up an era after an exercise of more than 15 years, is in fact has come into a very popular science. In the way back in 1995, the bacterial genome of approximately about 1.6 million base pair was in fact sequenced, and then it was shown that approximately 1600 genes were in fact are present in the bacteria. In 1997, the first eukaryotic genome was sequenced of the size of 13 MB in size, and in 1998, the animal genome was in fact sequenced approximately harboring about 20,000 genes. In the year 2000, the human genome sequence, a draft version of it was in fact put into the public database in such a way that it was indicated to consist of approximately about 3 billion base pairs, and which could contain approximately about 30,000 genes which are essential for maintenance of a human life, which in fact came into a popular science as well as a specialized science and became very popular. So, it was in the turn of the millennium that the major achievement which was achieved due to a composite efforts of a number of laboratories, 5 different countries, to be able to understand a 15 years of effort of more than $500 million worth of effort, wherein the blueprint was in fact first draft sequence in fact was published in the year 2000, and then was put into a public database, so that it becomes usable for the various scientists to make use of the information and then use it for appropriate purposes that are related to human health in general. So, in 1990, the Human Genome Project was in fact started. In June 2000, the working draft was in fact published. In the February 2001, the whole sequence was published, and in April 2003, the complete human genome spanning approximately about 99.9% of the sequence was put into the database. However, it could never in fact accomplish all the 100% of the genome which could be enlisted, primarily because of that 0.1% which makes 2 individuals different, and that is why the whole genome sequence to the 100% composition cannot be accomplished or cannot be completed, for the very simple reason that 2 individuals vary in their genomic composition of about 0.1%. This is the kind of a machine which was in fact used for more than 15 years to accomplish the completion of the Human Genome Project, wherein we have a large room which is filled with the sequencing, manual sequencing machines was used, and then the whole range of sequencing was in fact completed in the specific laboratories in the 5 different countries, and then it was assembled together and submitted into the public database which is currently being used. So, as of now, the Human Genome Project, which in fact got completed, took about 13 years, and for the Venter's genome sequence, which was published in the year 2007, which took about 4 years, and the Jim Watson, who discovered the structure of the DNA, whose genome was completed within about 4.5 months. As we speak, the efforts, the cost and the time which takes to identify, understand the human genome has been steadily coming down over the last 10 years, and as we speak it hardly takes about 1 day to complete the human genome sequencing of the project, wherein you hardly need about 1 scientist to accomplish this job, and cost of the sequencing as we speak is now coming down to nearly $1,000 or roughly about 50,000 rupees, with the coverage of the genome of approximately about 20 to 25 times, and then it can be accomplished by a single institution. So, the goal of being able to translate the genomic medicine, genomic sequences into the benefit of the humans and to be able to use in the genetic medicine is being realized, and in the coming years we will be able to see much more and more advances and the benefits of the genome sequencing to be able to bring to the practical use. What is shown here is the anatomy of the human genome as we understand today. As we have already talked about it before, the human genome is compartmentalized into 2 different organelles within a given cell. There are DNA which is present in the nucleus, and the DNA is also present in the mitochondria. The DNA which is present in the nucleus in fact encodes for about 28,000 genes. Out of these, approximately about 1400 of these genes are in fact code for mitochondrial gene products. These 28,000 genes are in fact present within roughly about 3% of the human genome, and the rest of the 97% of the genome do not contain genes. However, in fact 70% of the genome is transcribed into RNA, and nowadays we understand that in one form or the other these RNA are beneficial in maintaining the structure, the function, the integrity and the homeostasis of a given cell or a given tissue or a given organ. In fact, majority, roughly about 40% of the genome consists of what we call as the repeat elements. These repeat elements reiteratively occur within the human genome, and these are called lines, which stands for long interspersed nucleic acid sequences, signs, short interspersed nucleic acid sequences, pseudogenes, which are nothing but remnants and signatures of the DNA sequences that in fact are present in our ancestors, and the retro elements which may have integrated at one time or the other into the human cell. Every single cell has nearly about 3 billion base pairs per diploid cell, and every single gene has 2 copies within a given cell. As we understand today, one of the benefits which we have attained after the Human Genome Project is being able to understand how many genetic syndromes or genetic diseases which in fact are present or which in fact cause deleterious effects to the humans. A major effort which is started by Victor Makusek, the father of modern genetics, who has systematically started cataloging the genetic disorders in man in early 1960s, in fact have indicated that there are approximately about 13,000 genetic diseases which are in fact are manifested in the human beings or in individuals. More than half of them, about 6,000 of them are in fact are monogenic disorders, that is they are single gene disorders, and then rest of the nearly about half of them are polygenic disorders, or those disorders which are caused by multiple gene defects, which include, for example, the cardiovascular diseases, the asthma, the arthritis, the diabetes and so on and so forth are complex polygenic disorders which are part of about the 13,000 genetic disorders which have been catalogued. Most of the monogenic disorders in fact follow Mendelian principles of inheritance, wherein they are stably transmitted from within the pedigree from generation to generation. Mitochondria on the other hand codes for about 37 gene products, which include 2 ribosomal RNAs, 22 tRNAs and 13 peptides which are intimately associated with the oxidative phosphorylation or the respiratory processes that are happening within the mitochondria. The mitochondrial genome is approximately about 16.5 kilobases in size, and each mitochondria may have more than 10 copies of the DNA. At the same time, each cell can have more than 50 to 1000 copies of mitochondria, thereby contributing significantly to the bulk of the DNA that may be present in a given cell. In fact, they are represented, depicted in the form of homoplasmy, and homoplasmy identifies each of them as identical set of nucleic acid, and because of the lesser repair of the mutations that may be present in the mitochondria, they accumulate mutations at a much faster rate. They have limited repair capabilities, and as we know, unlike nuclear DNA and the 13,000 diseases, mitochondria approximately has about 100 syndromes and the mutations which are associated with that happen in the human beings. So, it has become a kind of a fashion to say that anything which we do is in fact is genetic. In fact, the robber also can say that, look, whatever the crime which I have committed is because of my genome, and it is not me who did it. And that's the kind of a trend which we are seeing, and that which we are evolving right now. I hope I have been able to tell you something about the advances that have been happened in the last about 10 years, and I hope that you have benefited from some of the information which we have given to you in this last half an hour.